Hi, I'm Oana Marcu, and hopefully this will show up soon. I happen to be one of the people that Mr. Wright introduced at the beginning as one of the people who waits 18 months for a grant to be approved. And you never know when you get a result, but to tell you the truth, actually the reason I do it and I still do it and I still want to do it is because it's really fun. So I work at the Carl Sagan Center at SETI Institute, which is 2 streets away from here on Bernardo Avenue, and I also have a lab at NASA Ames. And I'm a biologist by training, and unlike Carl, who didn't know coming out of high school what he wanted to do, I knew exactly what I wanted to do. I wanted to study behavior, to learn Latin, and to be a photographer for National Geographic. So I'm here now. So it's better not to know what you want to do. My first interaction with suborbital research came actually from the first suborbital research workshop, which was held in December 2008 in San Francisco in one of those hotels. And you won't be able to read here, but it says the purpose of the workshop was to bring interested researchers from a number of fields together with suborbital vehicle operators to exchange information and discuss the scientific potential of these exciting new capabilities. So for me, it's really, really neat to be here 4 years later and to realize that the research opportunities have opened up to you. Research is no longer held in a lab, held in a box. So there's a lot more opportunity now. There are a lot more possibilities. So it's actually really, really neat to be here. And what I wanted to do today was to talk a little bit about microgravity. Sean Casey asked me to talk about what microgravity really means. And simulating, what capabilities do we have to simulate microgravity on Earth? So Carl talked a lot about microgravity on the space station, which is actually days to weeks of microgravity to months of microgravity. But it's very, very different when you are on a suborbital flight and you only have minutes of microgravity. So what I wanted to do is give you a panel of types of experiments, and because I'm a biologist, I'm going to stick to biology, and ask the question, so you can see why this is actually, I think it's valuable. So what do suborbital flights add to what we already have? And what kind of useful biology can we do? And the useful biology, actually, I like to think it in 2 ways. Carl mentioned before that on the space station, if you there are a lot of deleterious effects of microgravity, like skeletal, muscle loss, immunity problems, and so on. So one of the useful biology we can do is to understand what the unwanted effects of microgravity are so we can counteract them. And the other one is everything is new and open, and microgravity might have really new beneficial outcomes that we actually don't know about. There are new processes that happen in microgravity and that we don't understand. And the reason we don't understand them is because we constantly live on 1G. That's all we have, and we can't get away from it. But if you want to go and explore into space, here's the G level on the moon, and here's the G level on Mars, and this is not, this is sort of to scale, the station is not to scale. So what we're interested in actually is a gradient. It's a gradient of the gravity vector, right? We want all this level of gravity and what's in between, which is 0. So because we constantly live on 1G and we can get away from it, it's really hard to understand what the effect would be. So the one way people do it is change the vector, go the other way around, and do hypergravity, which is usually done by centrifugation. But there are also really clever ways that folks have come up with simulating microgravity within the Earth's gravitational field, within our own boundaries. So I was just going to go a little bit through each one of these, kind of go through what the residual gravity is, and the time lapse is here, so you can see how that relates with some suborbital flights. So there's cleanup stats of bioreactors. So this is also a scale of size, from the size of a flask, a vial that you can hold in your hand, to huge magnets, to drop towers that are 100 meters high, and to parabolic flights. And I didn't put here sounding rockets, but you probably guys know more about that than I do. So just quickly, people at the Johnson Space Center were really interested in trying to understand microgravity effects on cell cultures. And Carl talked a lot about what biology and what areas of biology, but just think of cells, and it's easy, right? Pick up your hand, and we're all made of biology, so it should be of interest to everyone. So the thought was, okay, how can we think of microgravity and cell cultures? What would a cell culture need to experience on Earth in order to mimic microgravity. And obviously a cell culture would have to be in solution, in suspension, right? So people started developing these flasks with a magnetic stir or stirrers, but the problem is that these stirrers create a lot of shear stress. And actually in any kind of microgravity experiment, if your experiment, if your biological experiment is in a fluid, you need to think about what else happens to the cell, not to the microgravity itself, but to the fluid or liquid it is suspended in. So fluidics is really, really important, right? So then trying to get away from the shear stress, what they've done is turn these things around and put them on a rotator, and what they have come up with is the most commonly used device to mimic microgravity on Earth. And it's called a rotating wall vessel, and there are many different types. One is the high aspect ratio vessels, and this is a picture that we have this in the lab and put it in the incubator. It's made by a company called Syntecon in Houston. And the high aspect, it means that this is a very flat field with very low shear of cells. But if you have a cylinder that you'll see in the next picture, it's just called a slow turning lateral vessel. So I'm not going to go through this a lot, just to give you one example of where this could be useful. Again, as I said, in a lab, in a culture, if you have cells in a flask, they'll grow in a layer, right? You have the 1G vector that keeps them always settling down. If you continuously rotating them in this cylinder, what ends up happening is really fascinating. So the structure, from having it settle down and making what we call an epithelium, which is basically your skin, which has many layers, these things round up and make these three-dimensional cultures. And if you've heard about this, the promise of the 3D cell culture is that we're going to be able to reconstruct organs. Right, so now people who wait for months on waiting list for organ transplant, maybe they could benefit from something like this. So this is something that's very unique to microgravity. It's fascinating, it has a huge promise. So moving on, another way to simulate microgravity, it's kind of cool. So University of Nottingham, for example, developed this magnetic levitation. The thought again is how do you keep things in suspension, in the suspended environment of microgravity? They have this tremendous huge electromagnet that produces a field of 17 Tesla, and it's in a cryostat, in liquid helium basically, that cools it down to 4 degrees Kelvin. And it's a huge cylinder at this temperature, but in the middle it has this core that's exposed to air, and that's why you settle your experiment. And basically in a diagonal section, sagittal section, this would be the center, this would be the center of your solenoid of your magnetic field. If you put your experiment in there, what you're getting is 0G, or a simulation of 0G. And that's where the residual gravity comes in. So when you think of microgravity, it's not 0, it's less than 0. So, 10 to minus 2 or whatever it is, and I have those numbers there. So in the same experiment, depending on where you position within the magnet, you're experimenting, you get a mimic of 1G or 2G. So this is an experiment that was done with fruit flies on the University of Nottingham. So they were trying to put these flies into a magnetic field. These are all the gravitational directions and how they cancel out, then this zone would be 0G. So they kept this experiment. The advantage of it, it's expensive and difficult to do an experiment on the space station, but it's pretty cheap to stick things, not incredibly cheap, but it's more accessible to put things in a magnet. So they were able to repeat the experiment many times, and to do it for a different duration of days so they can see what changes. And one of the things they were looking at, Carl talked a little bit about the genes. I don't know if you're familiar with that, but things that do the job inside our bodies. And they found, for example, these panels, these genes that change with magnetic levitation in what they call mimic gravity. Now I happen to work with fruit flies on a space experiment on the shuttle that actually stayed on the middle of the shuttle to the ISS in 2006 for 12.8 days. That was on STS-121. And we published the data before, and what we also actually did the same thing. The experiment was designed to look at immunity changes in microgravity. So a lot of we use the fruit fly as a model for immunity changes. But of course, when you do an experiment, and this is important for you to know as well, expect the unknown. When you're going to fly your own experiment on microgravity on a suborbital flight, because no one has done it before, presumably that's how you're going to do it, be prepared to expect the unknown. and design it so you can look at everything possible that can come out. And that's exactly what we did with our shuttle experiment. We're prepared to look for the unknown, and we also designed, besides the fact that we're designing it for immunity, we wanted to look at everything that we could. So we looked at the entire panel of gene in the organism. So of course, when I first saw this paper, actually just like last night, I pulled out my old data, and I looked at, okay, intermagnetic levitation that mimics microgravity, does it have anything to do with the real spaceflight conditions that are on the ISS? And here's a gene that are on the line called peroxyredoxin, which is actually reducing the hydrogen peroxide, that is the same, and none of the others are. So what does that mean? So this brings me up to how do you interpret your experiment? So this was actually a very, very elegant experiment and very involved with really good statistical data. So right here, I put again for you to understand the design of a biological experiment. The spaceflight, of course, has a more accurate representation of microgravity. And you could also do measurements in real time. Depending on what you're measuring, you can actually do them on board of the ISS, so within microgravity, without having to wait for the return. If you have to wait for the return, one thing that happens, it's gonna happen on the suborbital flights as well, you're gonna have interference from hypergravity. And if you recall the flight profile that was put up for the length, when you launch and when you go back into landing, you're going to experience hypergravity, 2.8 to 2.4 G. So when you do your microgravity experiment, you want to make sure you measure or you have a control that measures for hypergravity. So that's an important thing to bring up. Of course, for a spaceflight, it's difficult or impossible to repeat, and it's more expensive. Magnetic levitation or any other kind of microgravity experiment that is on ground or more accessible, you can, this is really, really important. The N number can be high. You can repeat your experiment until you're sure you can get it right. And with a link flight, like 4 flights per day, that's a huge advantage. The problem is that results are more difficult to interpret. In this particular case, there's also huge interference from magnetic field. So their magnetic field, when I looked at the numbers and I compared with the Earth magnetic field, is 6 orders of magnitude. So the Earth magnetic field is micro Tesla. This was 17 Tesla. So there are things that we need to worry about. And the measurements are done only after exposure. But it's neat, and the fact that you see the same, one of the G's happening, it's giving you a clue. So you always have to be prepared and make the best educated guess that you can and be prepared for your experiment. That's the point there. Another type of microgravity mimicking is the drop towers. And I put here, there's one at the Glenn Center that provides 2 seconds of microgravity, but there's also one of the Center for Applied Space Technology and Microgravity at the University of Bremen in Germany. And Brad Bipal, my colleague who sits back there, said he used to ride his bike around it. So this is a 146 meter high tower with a 110 meter drop, and one drop provides 4.74 seconds of microgravity. Now you can see the residual microgravity in this case is actually less than we had before, so that's good. The problem is that it's short. It's short and non-repeatable. It's short. So what the Germans have done, I think since a few years back, they've also developed a system where they actually catapult from the ground, from the base up, and then coming down, and that doubles basically the length of the tower, doubles the length of your experiment. So it's kind of neat. This is the profile of microgravity. The type of experiments that you can do, is it useful to have seconds? Well, so if you want a biology experiment, if you are in physics and you can think of microfluidics, of course you're going to see changes. In biology, are you going to really see changes? Isn't biology more resilient than that? And actually, yes it is. Because if it weren't, we wouldn't have pilots that would be on ISS or would want to fly the Lynx plane, right? But there are things that you can look at, and some of the fastest biological processes that can happen are at the level of the membrane, either electron transport at the level of my membrane, or this is a membrane. This is, just imagine this is a cell, and actually this happens to be one of the cartilage cells that you can find anywhere in your body. And all across the membrane there are these pores. Well, if you think of microgravity and suspension or things that happen in microgravity, one of the things that you expect is extensions and contractions of the membrane would change the configuration of the pores. Well, these pores are actually passive, for passive transport. And what they transport generally is ions. Well, ions tend to be some of the things that induce processes to happen later in cells. And these things happen in the level of, there's a graph here that I've taken from a paper that came out in Biochemical and Biophysical Acta just recently, that's looking at these four that exchanging potassium and protons, that these things happen in nanoseconds. So what they do is measure conductivity, voltage, or the conductivity at the level of membrane, and these are things that happen within seconds. So that's cool, yeah? There are things that you can measure. And here's the design of an experiment, and of course this is paths for being dropped out of a tower, so it's a very different type of configuration. But this could give you an idea of what would be involved into an experiment that you could design for a suborbital flight. As I said, then you get other seconds of microgravity in parabolic flights. And the neat thing about it is, they are very, very short, but in this case, they are repeatable. Right? You get about 30 to 40 parabolic flights, so you get 30 to 40 seconds multiplied by 20 seconds of microgravity. Again, the same problem here. You have hypergravity before and after the flight, so that's a problem. But it's useful, and what these things are generally used for are a lot of experiments that involve either things in the cytoskeleton, which is the structure that right below here, right below the membrane, in order to keep this shape, each cell has a skeleton, just the way we have a skeleton in our body that keeps our shape. Each individual cell has what we call a cytoskeleton. Or for large gene screens. So this group, for example, this is an example that I took from one of the papers that uses suborbital flight. What they were looking at, this is the type of cell that you see in red, the nucleus, and in green you see genes or proteins that are associated with the cytoskeleton. So this is normal 1G and this is 0G after, in this case I think they saw the changes after 20 parabolic flights. And this is a significant change in vimentin, one of the proteins that's under there, right? So you can actually see changes. Now the question is, of course, do they mean anything later on? And the hope is that they mean something, but not too deleterious, right? Because you don't want to have too many effects. But that's up to you to decide what you're going to look at. So this is, for example, how they pack their experiment, and this is some of the stuff that Carl showed you in much more detail about syringes, the microfluidics, and flasks that you can use. And these are all not terribly expensive, commercially available, so anyone could design that experiment. Also, the reason I put this up here is because, as I said, there are things that have already been done with interesting questions. So when you design your experiment, think about what do those minutes of microgravity, what can those minutes of microgravity show me? So are they useful? That's why I put up this question. Are 4 to 6 minutes of microgravity useful for biology? And here I put a little bit of the time scale and the spatial scale for biological processes. And when you talk to people about gene expression changes in a cell, they always default back to this 20 minutes for one round of transcription in bacteria. What does that mean? You need 20 minutes for a gene to make more of the same thing. So this is DNA, for example, and this is when it's making copies. And this is where it's making copies of itself, which will make those proteins that Carl was talking about. So it takes 20 minutes for that, right? 20 minutes, we are only at 4 to 6. So hours for gene expression, and then days and weeks for physiological responses like growth and metabolism. So does it make sense to use 4 to 6 minutes of microgravity? I showed you before where it's useful to have seconds of microgravity because you look at fast processes right of the membrane. And then within minutes, what you can see are things that are, here's the key word on this slide, signaling. Signaling, in order to get to those 20 minutes, it takes from here to here. Whatever happens, it takes the order of minutes, right, within minutes. So this signaling involves molecules that are small, fast binding, fast diffusing, and really produced really quickly. So things do happen really fast in cells, right? And I put in here because I know we're talking, it's a microgravity workshop and we're talking about microgravity, but payloads on the outside will also experience radiation. So some of these signaling molecules, turns out life is tricky. Life adapts to changes in the environment. And because I work in astrobiology, we work a lot with extreme environments and things like that, you know, resistance to radiation, resistance to desiccation, resistance to salinity, all sorts of resistances. And life keeps adapting to all of those things. And so life, what something is perceived as deleterious to the cell, cells are really smart, life is really smart, and it turns out into something really good. So probably the way this signaling evolved is that initially they weren't very good and they were breaking up the DNA. And then they became signaling molecule. Now you can turn that around and say, well, if I already know about the signaling molecule, some of these molecules have a dual role in cells, and you'll find a lot of that. So and that's the key to this, and that's the key to microgravity research. Remember when I was telling you some of the things are really good and some of the things are not so good? Depends on how long you expose them to microgravity, how frequently, how long, how many times you repeat. So things that might break the DNA, so this path can go either for some making more proteins that rescue the damage, or they can just go to damage. So that's one of the most fascinating questions in space biology. How do you balance this thing between being exposed both to microgravity and radiation, and can you counterbalance any effects that you see? So basically, with 5 to 6 minutes of microgravity, what you're going to be able to do is go back, and you're not probably going to be able to address, bone loss or muscle changes, but you're going to be able to go inside the cell at molecular level and ask the fundamental questions about what's the first molecule that responds to microgravity. And is that useful? Yes, tremendously useful in my opinion. Right? We don't know what the long-term effects are, but for you can, because your experiment is going to come back. You're going to be able to do experiments later. But it's going to be a huge panel of things that we really don't know about, and we don't have the power, we don't have the capacity to do all that, we don't have the number of scientists. I don't want to put my graduates on all these experiments, and we just don't have the breadth to do all these experiments. So it's fantastic that citizen scientists come in and help. Then I put here, I gave these examples for microgravity and what you could do, but also here's one example of the tree of life, started from an ancestor that we don't know about, and going into the bacterial domain, archaeal domain, and what we call eukaryote. You and I are eukaryotes. These are more primitive organisms. These are some of the most important organisms on Earth. And they are of interest both to space biology, to all the effects that you have seen, but also to astrobiology. So you have that $10,000 prize is to capture organisms, but the key is what are they going to be, where are they going to fall on the tree of life, and what are they going to do? What do they do? Where are they? So these are, for space biology is interested in life support, like food, oxygen, energy, that all these microbes can produce all these in byproducts that are being made. If you make this transgenic E. coli, for example, that produces insulin that Genentech has done a while ago, and new processes. And this is the fascinating thing. You have a chance to look at anything that's new that we don't know about. So I put in here, how much more time?
Getting to the bottom.
Okay. So I put in here a few examples of organisms. This is called Ichthyostylum discoideum. It's called a slime mold. It's actually an amoeba. It's a very, very simple organism. I thought I had a better picture of it. Right here. That's kind of falling in between plants, fungi, and animals. And it's an amoeba that's unicellular, but it aggregates and it forms this multicellular organism. Now, this is one of the processes that you expect that microgravity would be affecting. At least I would be one who would want to fly this organism. And not only that, but NSF and NIH have just picked up on this organism, and a fantastic tool for screening for drugs and targets for drugs. So drug screening, these are some of the examples of drugs or molecules that have been screened for or looked for targets in the Ichthyostylum. And also a lot of fundamental biology, and if you look at all this, chemotaxis, motility, adhesion, growth, cell differentiation, signaling, these are all things that we very much expect to be affected by microgravity. So when you pick your experiment, try and think of what's the experiment that's going to show me the coolest thing that I expect would happen. Another one is algae, and Carl mentioned plants for food supply or not. Algae are also on the side of this. And these are slides from Brad Viva, my colleague who's sitting back there. They do this experiment with something called, they measure photosynthesis in space, or photosynthesis in the lab right now. So you can do it with this, it's called a PAM fluorometer, pulse amplitude modified. Basically, it's measuring the electron transport chain in chlorophyll. So it's measuring the efficiency of photosynthesis. These are the microtiter plates that Carl was talking about, so this is a good use for them. You can screen in a 96 well plate or 384 well plate a bunch of strains. You could screen for a bunch of strains that change photosynthesis in space. This is experiment from my own lab where we're looking actually at heat stress. But this is what you would normally see with no change in photosynthesis at all. And this is how you see declining photosynthesis in different conditions. This equipment is commercially available, commercial off the shelf. So this can be bought and included in to an experiment. Here's an yeast, which everyone knows about. It's been used for many years. And you can look at growth of survival, for example. So it's also important to choose your measure. You can do a complicated experiment, you measure all these things, but the experiment can be confounding. But you can also choose things to measure that will tell you a lot of things, but without being complicated. So growth of survival are really easy to tell. And there's also an experiment with Pharmasat here at Ames that flew this on a payload on a small satellite, looking at antifungal things using the Saccharomyces. So if you want to work with this, go check out these things. Things have already been done, have been compacted. They're not necessarily low cost. This is an example of bacteria. In this case, it's just the regular Escherichia coli. It's green because it carries a green fluorescent protein, which is the marker. You can have markers. It, again, bacteria can be used for microbial fuel cells, vaccine antibiotics. Something very new, synthetic chemicals. I can't exactly remember the university, but they were able to actually insert into the proteins new amino acids with a structure that doesn't exist in the real life. So this is a huge promise for something that is new, that we don't know about. And you can look at gene, and people have looked at gene expression. This is a GeneSat payload that flew out of Ames, and this is an antimicrobial, this is actually being made right now. It's ECAMSat, it's looking at antimicrobials in E. coli, and it's a collaboration with people from Stanford and Ames that builds this nanosatellite. So this is the 3U, or actually in this case it's the 6U cube version. So again, if you want to use bacteria, there are things to do. So what's really the key to me here, the promise is that you can do large screens with repeatable number of things. Yes, it's short microgravity. Yes, you're going to have to control for hypergravity, so make sure you have a centrifuge so you can repeat experiments without the microgravity, but with the hypergravity that the profile of the suborbital flight is going to tell you. And then you just have to subtract experiments. But one thing that I was talking in the break with someone here in the room, I was saying, you know, it's citizen science, but it can be really good and helpful science, as long as you design your experiment carefully. Look for what's available, look for what the information is, build your hypothesis, come up with something new, or build up on something that's existent already, and try to think, how is my experiment going to help the next stage? And because, you know, why limit yourself to suborbital flights? Next time you're gonna fly somewhere else, higher up. So I like to think in steps and building things for something bigger, you know, eventually a real flight in microgravity, in real microgravity. So enabling gene expressions, I'm not going to go to this because we're running out of time, but basically what this is something called polymerase chain reaction, which is the thing that changed, you probably saw it in Jurassic Park when they amplified out of the DNA from mosquito, the dinosaur, whatever that was called, PCR, polymerase chain reaction, it's amplifying a lot of DNA. And what you can do, the new technology called quantitative PCR is what measures the gene expression. It tells you in space. So for me, for example, that would be really fascinating to know in microgravity. And you have the advantage of so many genomes known right now. You can pick an entire genome and screen for it in one suborbital flight or repeat it. So that's pretty cool. So these are the kind of experiments that you can do. Carl showed you a lot of the hardware, so have fun. I also wanted to mention that this is not necessarily, I mean, we are really excited as researchers. We're trying to get away from being complete nerds. And we have opened up the doors for a long time. And I wanted to share a little bit, if this loads. Well, with my colleague Brad Lieb out in the room, we ran a science camp for students in science for 3 years. So hopefully the students who came to the camp are citizen scientists now, and they were part of our group working with us directly on what to build a mission for Mars. This is not for suborbital flight. The point that I'm trying to drive here right now is that science is accessible, and there's expertise available, there's hardware available, there's everything, so there's no reason for you not to do it. Students have done it. And there are some students in the room that I saw here right now. I see one right there, and I'm curious why you're here. But hopefully we'll see soon experiments from you in space. So yes, it's possible, and we're trying to help. Thank you.